Hi, my name is Ariel Hippen, and my poster is called Efficient Domain Adaptation for Cancer Mutation Prediction. Supervised machine learning focuses on building prediction models where patterns learned in the input data are used to predict the label. These kinds of models have been widely used in biomedical research. A standard caveat of supervised machine learning is that the classifier can only be used on data derived from the same distribution the model was trained on. This helps to avoid extrapolation. However, challenges in data acquisition often mean few or no labeled examples are available for the distribution of interest. Sample sizes may be insufficient to train on rare cancer types, for instance. For situations where labeled target data is unavailable, the field of domain adaptation has established principled ways to develop predictors for the data of interest, here called the target data, using labeled data from a related but distinct distribution, here called the source data. As an example of a use case for domain adaptation, we can think about predicting prices for motorcycles, the target distribution, using only data for cars, the source distribution. A feature like speed may be highly correlated across both cars and motorcycles. Gas mileage may have similar directionality, but with a weaker correlation in the target distribution. Other features, like number of doors, may not be transferable at all to the target distribution. A good domain adaptation method will build a classifier that prioritizes the most transferable features to make its classification. One domain adaptation method that was published in 2019 is weighted elastic net domain adaptation, or WENDA. WENDA is designed to specifically leverage the complex interactions that exist in genomic data. It infers that features that have a similar correlation structure to other features in both the source and the target data sets is likely to have the same effect on the feature of interest in both, and is thus prioritized to maximize transferability of an elastic net classifier. WENDA has three major steps. First, for each feature in the source data, it trains a Gaussian process model to predict feature F using all the other features. Next, it uses the trained feature models to predict the value of that feature in the target data. It calculates confidence scores for each feature based on how well the source model predicted the target values. Finally, it converts these confidence scores into a weight matrix, which is incorporated into the penalization term of an elastic net model, trained on the data and labels from the source distribution. Thus, the elastic net model trained on the source distribution is optimized for its transferability to use in the target distribution. My group was excited to use this method, but we found that generating a Gaussian process model for each feature was extremely time intensive. Fortunately, Gaussian process models are amenable to calculation on a GPU. We re-implemented the feature training in gPyTorch using LBFGS approximation. With these changes, running all steps of WENDA on a genome-sized dataset with over 10,000 features is now feasible in hours on a single GPU. Where training 12,000 models on 10 CPU cores reportedly took 51 hours for the authors of the original paper. When we tried to do it on 6 CPU cores, the model ultimately didn't finish and only trained 1,300 models over 68 hours. Our GPU implementation, on the other hand, ran all the models in 5 hours. To figure out exactly how much faster our GPU implementation was, we made simulated data sets with anywhere from 100 to 5,000 features and measured the time it took to train and generate confidence scores. For each feature set, WENDA GPU was between 7 and 12 times faster. Fast is good, but we also needed to make sure that our re-implementation was accurate. The prediction task in the original WENDA paper was predicting a person's age from tissue methylation data. On this data set, WENDA's GPU's predictions were highly similar to the original WENDA models, with a correlation coefficient of around 0.97. We found that with a regular elastic net, model performance is poor on data that has not been seen in the source data set, here the cerebellum tissue. But using WENDA GPU, cerebellum samples are predicted much better, even though the model never saw labels for this tissue type when it was training. Our GPU implementation of WENDA allows us to deploy domain adaptation to new contexts and bigger genomic data sets. For one use case we wanted to try, predicting mutation status of common cancer driver genes based on transcriptomic data. We tested if Wenda GPU would improve portability of classifiers of TP53 mutation status from one TCGA cancer type to another. Models were trained pairwise on 16 cancer types from TCGA, using one cancer type as the source and another as the target. We found that across all pairwise models trained with Wenda GPU more regularly outperformed a negative control with shuffled labels than the models trained with a regular elastic net did. For most cancer type pairs, our models trained with Wenda GPU had equal or higher accuracy than regular elastic net models. In conclusion, Wenda GPU is a fast and effective way to boost the predictive power of models in cases where labeled data is sparse or non-existent. This work is available as a preprint on bioRxiv. Thank you for listening to my virtual poster presentation.